Hi friends. Today we're going to cover cartilage and bone. It's relatively easy to show how they look on a microscope, but we want to go a little bit beyond that. We want you to understand. We want us to understand a little bit more why they look like that, why they are like that, so that once we understand, we don't have to memorize anything. We'll go to some basic things here right now. Remember that the body is essentially made of two compartments, the intracellular compartment and the extracellular compartment, which may include the vessels and everything. We know that the extracellular compartment includes mostly fluid, water that needs to be in an accurately, precisely well maintained. It's mostly by the kidney. But besides that, we have to understand that what makes the structure, what they give support to the body, are not really the cells. The cells don't give support. There are many of them, but they don't give really support. What does give support to the body is whatever is the extracellular matrix, what means that all the tissues and all the substances that are produced by the cells in the extracellular compartment. We mentioned, we actually remember that the fibroblast, which has a little bit of a spindle shaped, produced two things, right, for the connective tissue. One was fibers, mostly collagen 1, also other types of fibers, but this kind of protein is collagen 1. And we also have a ground substance, which is mostly sugars that give that kind of fill the blanks, if you want to say it. There's pretty much in the connective tissue, which is what I am reminding you what it is. We have a mixture of this ground substance and fibers, which are essentially excellent for traction. And we have to remember that all our tendons and capsules and ligaments, etc., are made mostly by collagen 1. And they're extremely tough as far as traction is concerned. But they are not good when it comes to pressure. If you compress a tendon, it will totally, fold down, if you want. Now, so when we go to cartilage and bone, we are actually dealing with two kinds of tissues that are produced by the cells, but they give us support. They give us phenomenal support. And we will start with the cartilage. And when it comes to the cartilage, I'll remind you, I'll try to put a brief summary here, reminding us the hyaline cartilage. Hyaline cartilage is the most important, and it is not just a fluid, it is something that is firm. The word, I'll write it down here for you guys, the name of the hyaline cartilage. The hyaline cartilage, the word hyalos means transparent like a glass. It would probably be more proper to call it like plastic, because actually hyaline cartilage almost looks like plastic. It looks pretty homogeneous, and it is going to be mostly basophilic. I'm going to try to make it like that. In other words, it will stain mostly blue with hematoxylin. Just pretend that it's all homogeneous. And it is made by the cells called chondroblasts, which then turn into chondrocytes. The chondroblasts are a layer of cells in the perichondrium, called in the outer layer of the cartilage, that almost look like fibroblasts. But then one then starts producing the cartilage, they get embedded within the cartilage. And these are the chondrocytes that live in groups, usually in groups of cells, each one of them in its own lacuna, if you want, in its own space. Now, it's important to remember that the vessels don't reach the cartilage. Cartilage has no vessels. So if we imagine here a vessel bringing nutrition, the products will have to be to reach these cells by diffusion, right? Now, one question you would ask yourself: how is it possible to have this degree of firmness that you have in cartilage? As far as we know, we do have, let's put here, fibers that are produced by the chondroblasts and are collagen type 2. But that doesn't explain enough. That those fibers of collagen type 2 are linked to the ground substance, which actually are to the, we have a link protein here, and we have a core protein, and we have a glycosaminoglycans, chondroitin sulfate, keratan sulfate, that are essentially attached to the collagen fibers. Actually, I probably should have drawn the collagen 1, the collagen 2 more actually thicker, so that we can actually visualize that the ground substance made of a mixture of proteins and sugars is tightly attached to the collagen 2, and on top of that gives you this basophilic appearance, mostly because of the negative charge of the glycosaminoglycans. This is the most common kind of cartilage, and you can find it in your nose, in the septum of your nose, in your larynx, in the rings of the trachea, and most of the articular cartilages of the long bones. This is the most common type of cartilage. Like I said, there's no blood vessels don't reach the cartilage, but if in the cartilage that is in the ribs, the cartilage attached to the ribs, we may have some vessels running in the center. Let me see if I can open this marker. So that, in other words, there may be some nutrition reaching to those thick cartilages. We're now dealing with with cartilage in general, and we're going to touch the subject of fibrocartilage. And fibrocartilage, for fibrocartilage, I will mention something like this. I will draw it mostly in red because the main fiber in fibrocartilage is the collagen type 1. And fibrocartilage usually appears eosinophilic, I mean red, because of the presence of the protein corresponding to the collagen type 1. Now, we have collagen 1 and collagen 1, and in between layers we have layers of hyaline cartilage. So we have a little bit of a mixture of hyaline cartilage and collagen 1, which gives it a very tough appearance. I want to write down here fibrocartilage. Fibrocartilage. Let's see if I can write here, fibrocartilage. The fibrocartilage is not in too many places, but it is in one place very, very, very common, which is in the vertebral bodies, between the vertebral bodies. This is if you imagine that these are vertebral bodies, you imagine that our spine cannot be totally firm. It has to have some possibility of bending laterally and forward and backwards. And so that's why we have here the fibrocartilage. And it has to be tough enough to support the spine, but you know, enough, movable enough to allow some movement if you want. Now, just for the sake of completeness, I'll mention to you that usually the central part has an area, a gel-like, which is the nucleus pulposus, can herniate usually backwards. And why, you will say, why would it herniate backwards and not forwards? Because there is a ligament in the front part that it won't allow this to happen. I believe that's a ligamentum flavum, if I'm not mistaken. Anyway, the nucleus pulposus will herniate backwards in those circumstances in which there is movement that should not be done. And they will start pinching upon the nerves and in the lower part of the vertebral bodies, like L1, L2, L5, L4. So that's what happens, and then once it pinches upon the nerves, you get that nerve, that tremendous pain that goes all the way to your legs. That is sciatica. Well, to finalize the issue of cartilage, we'll share with you what is the elastic cartilage. The elastic cartilage, I will draw it in blue for some reason. The elastic cartilage is a cartilage that is it is basophilic, like the hyaline cartilage. I'll write it down for you. Elastic cartilage. But the question is that not only does it have collagen type 2, which it does also the same similar type of glycosaminoglycans. When you look at it on high power, it looks like hyaline cartilage, but then when you look carefully, you see that there are some strands of elastin. So that means that this kind of plastic, if you want, or plastic-like substance is going to bend, is going to be able to bend like that, like you do it with your auricles, or other places like the epiglottis. Like, epiglottis is not that rigid, it's not that, it's not like a, it's elastic enough so that it can cover the upper part of the larynx. Also, some other components of the ear, such as the external ear canal and the eustachian tube, and some cartilages of the larynx. So you can see the elastic cartilage is not really that abundant. It's mostly in the area of the ears. Now, like any elastic tissue, when you look at it grossly, elastic cartilage may look yellow because of the elastic fibers are yellow on just with your naked eye. Also, you can stain the elastic cartilage with elastic Van Gieson or some other elastic stain, and the fibers, elastic fibers, will be very abundant and will stain black? Well, pretty much so far we have covered cartilage, showing you that they are, plaques, I would say, of plastic-like material that adapt to different circumstances. Now, we're going to talk a little bit about bone. And it's important that you understand how bone is formed and how much it is composed of collagen type 1, because it's essentially made of collagen type 1, but not only of collagen type 1. If it were only of protein, bones would probably be very soft. So we have essentially a group of cells that resemble fibroblasts but are osteoblasts that may become to produce bone. When they become to produce, they start appearing more basophilic, because they produce, since they are going to produce collagen 1, they are going to have more RNA, right? It's true. So, and once they start producing bone, they are going to start producing layers and layers. The first layers are a little bit usually in the beginning of the formation of bone, are kind of wavy, and it's called woven bone. And then those layers become more and more and more parallel, and essentially in bone, the layers are quite parallel. And if you would look at them in a three-dimensional form, it would, for example, this sheet would be made of fibers that go like this, and the other sheet would be made of fibers that go the other direction, and the other, it would be maybe of sheets that go in the other direction. So in other words, it's like a meshwork of parallel layers of bone. But then something is missing, and you know what is missing. In the formation of bone, the osteoblasts release something that we can call matrix vesicles. I'll write it down for you. And these matrix vesicles are going to are going to be deposited in different places where the calcium and phosphate salts are going to form crystals. In other words, we end up having crystals of hydroxyapatite in different locations of these meshwork and the different layers of bone. And so that's why bone is a very tough tissue that is so much resistant, even much more resistant than cartilage. So right now I'm going to draw a bone to give us an idea pretty much of what is bone like. I'll try to draw a humerus if I can't remember. This is the articular corpus. Let's something like that. Now, if you want to study a bone, you'll look at it, you'll notice that bones are very light. And actually, they support us all our weight, so that's a heavy weight. and they're still light. Now, you want to study bone, you want to have sections, and we have one trouble. You try to cut a bone with a knife, you can't, And you cannot cut it also with a blade in a microtome. You need to decalcify it. It's okay? You need to decal. You have to, in other words, you have to remove the calcium and phosphate from the hydroxyapatite. And what do you have left? Well, what you have left is the collagen type 1. In other words, the collagen type 1 is red like any other protein and like any other type of collagen. In other words, once you decalcify, you're going to see that the area of the epiphysis and metaphysis, there is a lamellar kind of bone. In other words, the bone is like made of little small bridges of bone making a meshwork. And the same thing happens to the other epiphysis and metaphysis here. And well, that's pretty much. Let us not forget that at the end we have hyaline cartilage. Otherwise, we will have to rub bone with bone, which does not be very pleasant. In other words, this is what we have. Now, what happens to this long part of the bone? Well, although the epiphysis and metaphysis are very, very strong for resisting pressure, they are not so strong when it comes to bending like that. In other words, here we have compact bone. Compact bone. You know that by experience, and usually we have some trabeculae in the center, so that the center is not totally, let's say, it's not empty. Besides, you know, especially the bones of the legs and the distal parts of the skeleton are replaced by fat. So we'll put here some adipose tissue. And also, let us not forget that the bone is the only source of blood, especially, you know, what we're going to see in the next chapter. The bone marrow is present in between the bony trabeculae, and so it is so important for us to remember. We do, of course, we do that, right? Now, if we want to remember the histology of the area corresponding to the epiphysis, it's not very difficult. In other words, you have, let's try to make it like that, we have different anastomosing bridges of lamellar tissue that are filled with bone marrow and have cartilage on top, and that is essentially the bone. But we have to remember one important thing. Bone, once it's especially when it is ossified, it's also calcified, doesn't allow very well the nutrients to go back and forth like you could have in a cartilage, in any of these cartilage, in these types of cartilage. We have to have a certain situation for that. In other words, when we have an area here corresponding to a vessel, the cells, the osteocytes, are stellate-shaped. The osteocytes are stellate-shaped so they can reach, and they are so good to each other, as you want to say, so that the osteocytes can also share the nutrition with other osteocytes here and there. So that's the way the bone is actually maintained alive. Blood vessels do get into the bone, but they don't get in everywhere. They know they go to specific places. Here, the blood vessels are pretty much in all this, inside this trabeculae, but the oxygen will not reach the cells that are in between. If you want to get, I have to put it here, these osteocytes, because they are calcified, and the osteocytes will have to reach to them and share with the other osteocytes. So these are the layers of collagen 1 that's going to be calcified. And the osteocytes are in lacunas, and they will have to share the nutritious components via gap junctions. Okay. Now, this takes care essentially of the epiphysis and metaphysis. What about the long part, the shaft of the bone, the diaphysis? You probably have an idea about it, but if you kind of want to enlarge this area, it would be easy to enlarge like this. And we will notice that the lamella, instead of forming bands, anastomosing bridges, form concentric layers that form a cylinder. And we have this is the canal of Havers. What is the importance of having a central hole in the center of this cylinder, where the vessels are here, and the nerves and the lymphatics are in these Havers, in these Haversian canals, if you want. Now, if you need to get additional vessels trying to reach this, we have another type of channels called the Volkmann's channels. Obviously, I don't need to tell you, but the osteocytes are going to be here. They are each individually in an individual lacuna, and each of them is like a little spider, if you want, reaching to the next one and sharing the nutrition and the oxygen. Of course, this area, you don't have only one cylinder. You have multiple cylinders of bone, like here, for example. Some of them are already reabsorbed, some other are not. And so you have multiple cylinders in different stages of formation. At the outer aspect of the bone, we have a layer of bone like this, which is the outer circumferential lane. And also in the inner portion, we have an inner circumferential lane. So in other words, this is a compact bone made of columns of bone, which are by themselves made of cylinders of collagen 1, which has been already calcified with deposition of hydroxyapatite. For example, here with this one will go in this direction. The other one will go in another direction, and the other one in another direction. So this is very, very tough and difficult to bend. And all these structures, all these lamella, both are strengthened by exercise. Exercise actually determines the degree of development of these trabeculae. Here, essentially, that's what we have in the. Obviously, we have to remember one component, which I will put it here. There's one cell called the osteoclast. osteoclast. Which, what it does is actually it starts chewing up pieces of old bone, digesting it, and the body produces more bone. So in other words, our bones are not static structures, but they are permanently forming and destroying, like all our cells or all our body, the osteoclasts. I have a couple of things also to tell you guys. It's not easy to remember, but I'll try to put it here. The bone is formed, doesn't appear in the fetus like bone, obviously. It may appear in the bone like a membrane or like a model of cartilage. So we have an intramembranous ossification and an endochondral ossification. When it comes from a membrane, for example, let's put it here. If this is for example, the parietal bone, essentially at the beginning it's a membrane made pretty much like made of primitive connective tissue, mostly like stellate-shaped fibroblasts, mesenchymal cells, that is foldable, and this is the kind of intramembranous ossification happens in the bones of the skull, mostly the vault of the skull and part of the face. Later, some areas of ossification appear in ossification points, and these areas also of ossification grow, and this bone is converted. This membranous tissue is converted into bone. Now, when you have a newborn and you touch the forehead, it looks like the fontanelle is still soft. That is because this membrane has not completely turned into bone. Now, the other issue is concerning the endochondral. This was the intramembranous ossification. The endochondral ossification means that this bone, for example, when the baby is small, will be pretty much like this, made of hyaline cartilage. In other words, as long as the muscles and the structures are being formed, this cartilage component is going to be there and then has to be transformed into bone. How does it happen? Well, we have an area in the epiphysis and the diaphysis, let's say, in which the cartilage begins to degenerate. The cartilage becomes bloated, dies, and it becomes calcified, and then vessels penetrate here, and this is the primary ossification method. The bone starts the thing is happening, advancing in both directions, and here happens with the same thing. Obviously, the cartilage will remain. The cartilage that is in the epiphysis is the one that remains on both edges. Now, what is obviously important to do is try to understand why bones grow in length. And in order to grow in length, there is let's quote it, a fight between the bone that tries to reach the cartilage and the cartilage that proliferates. You know, the cartilage is very basophilic and forms columns of cartilage that multiply very fast. The lower edge of the cartilage is converted into bone, and the upper portion proliferates very much and keeps proliferating after birth, after the person reaches 18, 20 years of age, and it doesn't grow anymore in height. But here we have the epiphyseal plate that is going to one moment close, and there will not be any more growth in length. So this is what we call the endochondral ossification. Let's click on endochondral. Plus the other one is the intramembranous ossification. Let's go here, intra. Okay, intramembranous, and the other one mostly in the long bones. We have covered most of the topic. There's probably something that you may want to know, perhaps what happens when you break a bone. When you break a bone, something like that happens. That I'll try to use this eraser, which doesn't touch it. So we have a bone that has been broken. That's pretty painful, huh? I know for those who have had a broken bone, that's not very easy to remember. Now, anyway, what happens when you break a bone is that we get a good amount of blood. Let's use a different color of red to show us that we have a large amount of blood being deposited in the area of rupture. Well, obviously, you're going to have a cast or some other means of holding both pieces together, which will allow the body to heal itself. And the first thing that the body has to do is to get rid of all this hemorrhage and all this tissue debris. And so we'll have macrophages, connective actually granulation tissue, and everything is cleaned up to a certain extent. After that, only after that, there is the formation of some kind of a cartilaginous bridge that joins both ends of the bone. And after that, which is actually the callus, the primary callus, it's still movable, it's not firm, it's like cartilage. And then that cartilage will be replaced by bone, by bone. But that's going to grow into the cartilage, and eventually both pieces will be held together. Essentially, this is what I wanted to say you, but also remind you one little thing. When you go to surgery, for example, and you have an incision, suppose this is the epidermis, and the surgeon has made a good incision in the you know that the sutures that are placed here help quite a bit. The sutures that the surgeon leaves, but they don't close really that well. What makes the healing of any wound, a surgical wound especially, is the fibroblasts of the connective tissue, the vessels that proliferate and form a scar here and close the wound. Okay, I have been lucky to be able to share with you some insights. I hope that you understand things. And now you can go to your books and read and enjoy and pretty much have fun. We'll see you next time. Take care.